All right. Moving on from protein synthesis to DNA replication. DNA replication. So our cells divide in varying rates. Skin cells divide very frequently. Stomach lining, it's very acidic, so it's replaced very frequently. Slower rates, things like bone. So whenever cells are duplicated or copied or go through cell division, the DNA has to be replicated first. And we'll look at mitosis and the various stages of cell division. DNA needs to be replicated. So this textbook that we have, the Saladin textbook, shows DNA replication in a somewhat simplified form. So we will stick to the simplified form. So let's say, I'll just put up some quick notes here. So there's a long string of DNA. I'm not showing it in the double helix because it'd be difficult to draw. This is DNA. And some of our DNA is very long. Our DNA stretches like, I think it's something like 1.5 meters or 2 meters or something. If you were to stretch out our DNA, we have our genome, by that I mean human genome, is 3 billion base pairs. 3 billion base pairs. That's a lot. That's a long string of DNA that needs to be replicated. Can you imagine if our enzyme, DNA polymerase, were to come along and try to do that from front to back, from one end to the other? That would take a very long time. Instead, what occurs is that DNA polymerase starts to replicate DNA at several spots. So here might be one. So this is the same strand of DNA. This is the point of origin of replication. And an enzyme called helicase unzips the DNA at these spots. And it does that at the point of origins in many spots along the DNA. So DNA is replicated in different places at the same time. What that forms are replication forks. So this area here let's see, I'll use black. This area here and this area here, those are replication forks, where new nucleotides are formed by complementary base pairing So a new strand is formed at the forks. I'll just draw one here. So DNA can only be added to one particular end of a new strand. So in this direction, as the DNA starts to open, this strand can be formed continuously, but DNA can only be formed in one direction. So when the strand opens the other way, it can only be formed in little pieces like that. And these are called Okasaki fragments. I think it's Okasaki. And they are attached by ligase. Ligase is an enzyme that forms bonds between the segments. So I showed you that so that when I show you this, the replication fork, just so that you know that there are two forks, but we're only showing one of them here, the replication fork. Yeah, and the polymerase that's responsible for building the new strand is called DNA polymerase. So here you can see it forming a new strand. And eventually, so there's a new strand forming. There's a new strand forming here. Eventually you get a new strand, and the old strand and eventually, when the process is completed, you will have replicated your DNA. And it's called semi-conservative replication. Semi-conservative replication because the old strands are conserved. So here is the DNA polymerase. One is here. It shows that the strands are being made continuously, but I just wanted to alert you that that is not necessarily the case because of the problem of forming new DNA strands in a continuous fashion. But for whatever reason, the textbook shows it as one continuous process on both strands. So there's plenty of nucleotides in our nucleus: thymine, adenine, cytosine. So they are there and ready and available to be pieced together. There are errors. Jen, that's good. Good comment. Yeah, there are errors for sure. Errors in mutations. In bacteria, there's 3 errors per 100,000 bases copied. But if that were the case, every generation of cells would have 1,000 faulty proteins. So what happens with bacteria and with human replication is proofreading and error correction. So a different enzyme, a polymerase, proofreads the new DNA strand and makes corrections, which ends up in 1 error per 1 billion bases copied. And since, if you remember the genetic code, the codons, like 4 codons, may code for the same amino acid, and the only difference will be in the 3rd nucleotide of the codon. So if that mutates, it doesn't make any difference. In other words, some cause no effect, but occasionally they can kill the cell. They can even turn it cancerous or cause genetic defects in future generations. To cause genetic effects in future generations, however, the mutation must take place in germ cells. So in the cells that are to become eggs and sperm. So how long is a cell around any given time? Well, that varies, as we saw. Skin cells are around for a short time, bone cells are around for a long time. But each cell does have a cycle, certain phases that it goes through. So the first gap phase is when the cell is growing, somewhat it's performing its function. So if it's a liver cell, it's detoxifying some blood. So that's just normal when the cell is functioning as it would normally. Then there's generally a signal that the cell should replicate, and the signal is a growth factor. When that signal occurs, then the DNA will replicate. And that's called the synthesis phase, S phase. The second gap phase is when the cell grows more, but it grows in order that the new cells, the daughter cells, will have enough cytoplasm and organelles to survive and become functional themselves. Yeah, so that's called the G2 phase. That's growth. And preparing for mitosis. So mitosis is called the M phase, the mitotic phase. DNA has already been replicated at this point. The G0 phase, well, that's when cells have left the cycle. The cell has died, essentially. Yeah, and the cell cycle duration varies between cell types. So mitosis is the several phases of cell division. And we'll look at those separately. Prophase. The chromatin, which is the DNA uncoiled, coils into chromosomes. And each chromosome contains 2 genetically identical sister chromosomes. So whenever you see a chromosomes that look like that, those are 2 identical sister chromatids. And this is the centromere here. That's where they're joined.
Maria, is it the top part, and is the mirror across the middle horizontally, or is it across the middle vertically?
Horizontally, yeah. So the length of that is 1 chromosome. And the length of that, that's another chromosome. Does that help? Yeah, so they're joined. Oh no, I erased the whole thing. Yeah, so this will be 1, this will be another one, and then they'll join at the center, like that. So that's prophase, and then In order for the cell to divide, the nuclear envelope, the envelope that surrounds the DNA, has to disintegrate. So that's a step in prophase. And the centrioles sprout microtubules, and they push them apart toward each pole of the cell. So that occurs here. So here are the centrioles, and they're starting to sprout microtubules. And these are the chromosomes there. So there again is a prophase chromosome and sister chromatids. A metaphase occurs when the chromosomes are lined up in the center of the cell. Meta means middle. They're lined up at the equator. And then the microtubules from the centrioles attach to the centromeres of each pair of chromosomes. The other end anchors to the plasma membrane. Anaphase is the phase in which the centromeres split into two, and each chromatid, each sister chromatid, moves towards opposite poles of the cell. They move down by kinetochore protein, dynein. It's a very similar protein that functions in a flagella movement and cilia movement. Telophase is when the chromosomes begin to uncoil, the nuclear envelope begins to form again. The cells aren't completely divided, but the spindles start to break down. In cytokinesis, the cytoplasm is divided. It overlaps telophase. So that is where it's kind of like a belt around your body. So a myosin, which is a contractile protein, pulls on the microfilaments of actin in the membrane skeleton. So it pinches off eventually in the middle, just like a belt would be getting shorter and shorter. It causes a crease around the cell equator. It's called a cleavage furrow, and the cell pinches in two. And at that time, interphase begins, which is the normal functioning of the cell. When do they decide to divide? Well, not until there's enough cytoplasm for both daughter cells. The DNA is completely replicated. There's enough nutrients for the new cells, growth factor stimulation from an external source. Often, if tissue has been damaged, there's open space. So that's a signal for the cells to divide. And they stop dividing when there are no growth factors. Perhaps there's a lack of nutrients, or the cells contact other cells when they've started to form. There are the other cells, and then they won't continue to grow because there's no room to grow into. All right, I'll stop there, and I shall